We call it the ocean. But it's not. It's a memory of an ocean. A graveyard for our convenience. For every disposable cup, every bottle, every single-use wrapper, there is a ghost here. A synthetic ghost that will never truly die. We measure it in tons, in micrograms, in parts per million. But you can't measure a broken world with a ruler. You can only feel it. And I've been feeling it for a very long time. The lab is a chaotic symphony of humming machines, glowing screens, and glass speakers. A person, their face etched with exhaustion and disappointment, stares at a microscope. Their once sharp eyes are now weary. Their assistant, a young person full of unspent optimism, is busy cataloging samples. Another one, the assistant says, Sample 47B, mostly polyethylene terephthalate. Just like the last 46. The usual suspects. The main scientist doesn't respond. They simply sigh, the sound more of a gust of wind than a human breath. They adjust the microscope's focus, their fingers moving with a practiced, mournful grace. It's not just the amount, they say to the assistant. It's the scale. We're not just dealing with bottles anymore. We're dealing with nanoplastics. The stuff that's so small, it can cross the blood-brain barrier. It's in the air we breathe, the water we drink. It's in us. The assistant nods, their smile fading. They know this lecture by heart. I know, but we're trying. That's all we can do. A humorless laugh escapes the scientist. Trying is what got us here. Trying is the scientific equivalent of holding a bucket under a waterfall. We need a new river, a new solution. They lean back, rubbing their temples. They've been doing this for a decade, and the despair is a cold, constant companion. Their gaze drifts to a jar on a shelf, a forgotten curiosity collected from a deep-sea trench a few days prior. It glows with a faint, otherworldly blue light. What did I even collect that for? The scientist mutters to themselves. A weird form of plankton? I don't even remember. They get up, move to the jar, and hold it up to the light. The glow pulsates, a living, breathing ember. It's beautiful, but also strangely unnerving. They had forgotten to label it. Oh, that, the assistant says. I thought it was just a strange new species of bioluminescent phytoplankton. It's lovely, though. The scientist squints. They take a small sample with a sterile dropper and put it on a slide. They place it under the microscope, their exhaustion momentarily forgotten, replaced by a flicker of scientific curiosity. They look into the lens. Their heart stops. The image on the screen is not a simple, glowing microorganism. It's something far more complex. The glowing blue cells are moving, but not randomly. They are swarming, congregating around a clump of microplastic fibers they had added to the slide earlier. And as they watch, the fibers, they're shrinking. No, not shrinking. They are being consumed, broken down. No, that's impossible, they whisper. What is it? The assistant asks. The scientist doesn't answer. They keep their eye to the lens, their mind racing. They increase the magnification. They see the microscopic cells attached to the plastic, their membranes dissolving the polymer chains, breaking them down into something else. They run a quick spectral analysis on the new substance. The results flash on the screen: CO2, water, and simple, non-toxic organic compounds. The very building blocks of life. It's eating it, the scientist says, more to themselves than to their assistant. It's metabolizing plastic. The assistant leans over, their face a mixture of confusion and disbelief. But that can't be right. There's no known organism that can do that. Not at this speed. Not with this kind of efficiency. The scientist looks at them, their eyes wide, a new fire burning in them. I know, they say, but the evidence is right there. It's not just a biodegrader. It's a biosolution. Weeks have passed. The lab is no longer a graveyard of failed hopes. It's a hive of frantic, focused activity. The scientist and their assistant are running a battery of tests, their excitement palpable. They've confirmed it. The unique algae isn't just a curiosity. It's a miracle. It thrives on polyethylene, polypropylene, even the notoriously stubborn PET. And its byproduct is completely harmless. They've tested it on dozens of samples of ocean water and contaminated sediment. The results are astounding. It's almost too good to be true, the assistant says. A single-celled organism that cleans up our mess. And the glow, it's like it's announcing its presence. The scientist smiles for the first time in a decade. The glow is just a byproduct of the metabolic process. A little scientific flourish. But you're right, it's too good, which means no one will believe us. They know the world they're about to re-enter. The world of corporate interests, political skepticism, and a scientific community that's slow to embrace the radical. They show the assistant a rough draft of their paper. The data is all there. The methodology is sound. We have to be meticulous. Every number, every graph. Because the moment we publish this, we'll be a target. A target? For whom? The assistant asks. The ones who got us into this mess. The petrochemical giants. The plastic manufacturers. The ones who profit from the status quo. They'll see this not as a solution, but as a threat. They're right. Their fears are soon realized. The scientist stands before a packed audience of colleagues, their heart pounding. They're presenting their findings. The slides flash on the screen. Graphs of plastic degradation, stunning time-lapse videos of the algae at work, and the final, clean water results. A renowned professor, a titan in the field of marine science, stands up. Her expression is one of cold skepticism. Your data is compelling. Almost too compelling, she says. Have you considered the possibility of a contamination? Or a misinterpretation of your spectral analysis? To claim you found an organism that can not only metabolize plastics, but do so with such efficiency is frankly preposterous. A low murmur runs through the audience. The scientist takes a breath, their voice steady. We have run these tests hundreds of times, Professor. We've used multiple controls. We've even shared blind samples with a third-party lab. The results are consistent. This is not a hoax. This is not a mistake. The professor scoffs. And what about the long-term effects? You've only been observing this for a few weeks. What if this algae has an unforeseen impact on the ecosystem? What if it consumes the phytoplankton that are the basis of the marine food chain? You are proposing releasing a completely new species into an already fragile environment. The risks are astronomical. The room is divided. Some look impressed, others deeply suspicious. The scientist feels the hope draining away. They don't see the solution. They only see the risk. A sleek, black SUV is parked across the street from the scientist's apartment. A well-dressed man, a corporate representative, sits in the back, his face grim. He's on the phone. He's a problem, he says into the phone, his voice low and menacing. This algae of his, it's a direct threat to our entire industry. I want a meeting with him, and I want him to understand the consequences of his actions. Make it clear that this discovery is best left undiscovered. The next day, the scientist receives an anonymous email with a picture of their assistant leaving their apartment. The message is simple: some things are better left alone. Their initial fear for themselves is now replaced by a protective fury. They call their assistant immediately. I need you to stay with a friend. Don't go back to your apartment. Someone is watching us. The assistant's voice is trembling. Who is it? The people who don't want us to succeed, the scientist says. They're not going to stop. We have to be smarter. We have to be faster. They know they can't just publish the paper. They need to prove it works on a global scale. They need a demonstration so undeniable that no one can refute it. The plan is radical. They found a small, heavily polluted bay off the coast of a remote, uninhabited island. They will introduce the algae there, a controlled, monitored, and highly public demonstration. They have invited a few independent journalists and a handful of trusted scientists to witness it. This is our last shot, the scientist says to their assistant, a map spread out on the table. We have to document everything. The water samples, the sediment tests, the video footage. No one will be able to deny it. But the corporate pressure intensifies. The corporate representatives' men try to sabotage their equipment, threaten their crew. The scientific community is still skeptical, refusing to fund their work. The scientist has to sell their own home to finance the expedition. What if something goes wrong? The assistant asks. What if the professor was right? The scientist places a hand on their shoulder. We've run the simulations. The algae is highly specialized. It only targets long-chain synthetic polymers. It doesn't touch the organic matter. We're not creating a monster. We're creating a cure. We have to believe in that. We have to bet on hope. The bay is a ghastly sight. A thick layer of garbage floats on the surface, and the water is a murky brown. The smell is overpowering. Journalists stand on the shoreline, their faces grim, their cameras ready. The scientist and their assistant, in a small dinghy, release a small, controlled amount of the glowing algae into the water. It spreads out like a shimmering blue cloud, a stark contrast against the dark filth. For the first few hours, nothing seems to happen. The journalists are skeptical, their expressions reflecting the same doubt the scientist has fought for months. One of them, a veteran environmental reporter, approaches them. "Are you sure this isn't just a glorified light show?" the reporter asks. The scientist, wiping sweat from their brow, says, "Give it time. Nature doesn't work on our schedule." The next morning, the change is subtle but undeniable. Patches of the water are clearer. The oily sheen is gone. By the afternoon, the transformation is a revelation. The water, once a cesspool, is now a deep, clear blue. The plastic debris, once visible, has been reduced to a fraction of its former self. A fish swims past, a glimmer of life in what was once a dead zone. The journalists' cameras are now whirring, their skepticism replaced by stunned awe. The scientist and their assistant watch the news reports. The story of the algae that eats plastic has gone viral. Videos of the bay's transformation are everywhere. The world is watching. The phone rings. It's the professor. "I have to admit, I was wrong." Her voice, for the first time, is filled with respect. "Your work, it's extraordinary. It's a paradigm shift." Her words are a balm to the scientist's soul. The scientific community is now clamoring to work with them. The corporations are calling, not to threaten, but to offer partnerships, terrified of being left behind. The world is finally listening. Years later, the research vessel is now a fleet of sleek, autonomous ships, releasing genetically engineered algae into the oceans. Drones fly overhead, monitoring the progress. The Great Pacific Garbage Patch is no longer a swirling soup of waste, but a vast, shimmering blue expanse. The algae's glow can be seen from space, a blue-green line of hope spreading across the globe. The scientist, now a celebrated figure, stands on the deck of a new, state-of-the-art research ship. They're looking at the water, which is now teeming with life. A school of dolphins leaps from the waves. The assistant, now a leading researcher, stands beside them. "It worked," they say. "It's working. We think we are the masters of this world. But maybe we're not. Maybe we're just the students. And maybe, just maybe, the answers to our greatest problems were never in a machine or a factory. They were always here, in the quiet, luminous heart of nature itself. We just had to learn how to listen, and how to let a little light in."